So today I'm going to deviate from the syllabus a little bit by delving into the reproductive system, but we're going to start with the male system first. Even though on the syllabus I said we'd start with the female reproductive system, I think I'm just going to follow the order that's in the book. And another reason is that the male reproductive system is a little less complex than the female reproductive system. So let's first talk about what the reproductive system is. Now, this is the only system that isn't really essential to the function of your daily life. It does affect other systems, however, but you have to remember that it doesn't really change the way you're going to go about your day-to-day business. It's not going to guarantee your survival. However, it does guarantee the survival of the species as a whole. That is, that this is the one system that we use to maintain the species, but we don't really need it ourselves. So we have the reproductive organs. These are the ones that are going to first be producing the germ cells, and then it's going to arrange the meeting between those two germ cells. And then once the meeting has been arranged, then we will have to have a place for this new individual to be nurtured for the first, well, in humans, first 9 months. We will see that we have, not only do we have specialized cells that are produced called germ cells, these are also called gametes, but we're also going to see that there are differences in hormones within these systems. Surprisingly, many of the hormones in both the male and female reproductive systems are the same hormone, the same actual compound, but we'll see a differential sort of production of these hormones and when and how they're released and the timing of these hormones. We will also see that these hormones do affect other systems in the body. All right, the gonads, these are the things that actually produce the gametes, and the gametes are the germ cells. These are the reproductive cells. These would be the sperm or the egg. The gonads also produce hormones, so this would be things like testosterone, estrogen, progesterone, and so forth. The gonads will be the testis in the male and the ovaries in the female. Then we're going to have all kinds of ducts that are going to transport these gametes somewhere. And then we're going to have accessory glands that will be supplying things, liquid secretions that might have, for example, in the male reproductive tract, fructose, things that the sperm will need for their survival and for their motility. And then we're going to have the external genitalia, which is the stuff on the outside that you can actually see. So this would be things like the penis, the scrotum, the labia, and so forth. All right, so the reproductive tract is going to include all the chambers and passageways that connects the ducts to the exterior of the body. So that is from the gonads all the way to the external genitalia. So we have both male and female reproductive systems, which obviously have to be functionally different. Males are producing gametes all the time, from the time they reach puberty even on to death. Even though there is a decline in the amount of gametes that they produce, they're still producing sperm throughout their life. Females will produce gametes typically once a month. Sometimes that cycle can vary. And basically we're going to have that cycle go on from about the time they reach puberty to about the time they reach 50 somewhere, typically. So we will see that they're functionally different, but nevertheless, they're both derived from the same embryonic structure. So both male and female reproductive tracts are derived from the same embryonic structures. And even once we're matured, we have some vestiges, most of us have some vestiges of the other reproductive tract. So we have what's called the Wolferian system, which is the male reproductive system, and the Mullerian system, which is the female reproductive system. And interestingly, the female reproductive system is the one that would develop by default. So in utero, there is a burst of testosterone, and that acts as a switch to switch on the development of the male reproductive system. And so if you have a baby boy or someone who's genetically male who does not get that in utero switch, that is that burst of testosterone, then they will actually develop as female. Though they won't be functionally female, they will be phenotypically, that is to say they will look female. And by all intents and purposes, you would think that these are females. And there are actually quite a few people in the population who are genetically male, who are actually phenotypically female. And it's a rare situation, but it does happen, and it's not unnatural. All right, let's look at the male structures first, because the male structures are the ones that are first introduced in the book, and they're also a little bit simpler than the female structures. All right, so we have the testes. These are the male gonads, and these are going to make the androgens, and androgen basically means maker of men. So these are the hormones that we associate mostly with males. Now, we have to, like testosterone, for example, but we have to understand that both males and females produce testosterone, both produce estrogens, and we'll get to that a little bit later on. It's a matter of degree, of amount. So these males are going to produce gametes, which are called sperm or spermatozoa, and then we're going to have the pathway of the spermatozoa from the testes through a structure called the epididymis, where they will remain and mature for a while. Then we have the ductus deferens, which is also called the vas deferens. Then we will have the ejaculatory duct, and finally the urethra. So if we step through each one of these, we can also see that along the way, we're going to see accessory organs that will be secreting fluid into these ducts. For example, we have the we have the seminal vesicles, which will secrete fluid into the ejaculatory ducts. In the urethra, we'll have the prostate gland, the urethra. Urethra basically passes through the prostate gland. We call this the prostatic urethra when it passes through there. And then we have the bulbourethral glands, which we'll see, and they're right sort of at the base of the membrane that separates the prostatic urethra from what we call the spongy urethra. And the spongy urethra runs through the penis, and it runs through a structure called the corpus spongiosum. Now we're going to have some accessory structures, because if you remember, the testes of the male are on the outside of the body, and this is to create a lower temperature environment than the body temperature, because sperm need to mature at a lower temperature than your typical body temperature. So the testes have to be outside the body, and so we have to have a pathway for things to reach the testes, like blood vessels, and we also have to have the ductus deferens reach the testis, or from the testis up through the passageways into the urethra. So the ductus deferens will pass from the testes up through these structures called the spermatic cords, and then they will then empty into the urethra at the base of the bladder. So basically they'll curve back up into the base of the bladder, and then we'll have the ejaculatory ducts. We'll be receiving some fluids from the seminal glands at this point in time, and then that stuff will end up in the urethra. So we also have to have a means for blood vessels. So example, the testicular artery has to go through and the veins, the nerves, lymphatic vessels, and so forth. We'll see a whole venous plexus called the pampiniform plexus when we look at the structures within the spermatic cords. And these are basically some connective tissue sheaths that house all this stuff that pass through what we call the inguinal canal. And if you recall, the inguinal region is the region where the legs join the torso. And so the inguinal canal is the canal that passes through the base of the torso down to the testis in the males. And females have inguinal canals too, but they're not nearly as vulnerable as the males, because the males have more structures passing through there. So the inguinal canals are actually a prime area for hernias to develop. So if you have an inguinal hernia, sometimes part of the intestine can get down into the scrotum. It can be somewhat problematic, and usually that has to be repaired. All right, let's talk about the spermatic cords a little bit more. These are going between the abdominopelvic cavity and the testis, as we've said before. They've got layers of fascia and muscle. They're going to pass through this inguinal canal. This is a passageway through the abdominal musculature, and once again, it's right for tearing and getting hernias so that parts of the intestine can get down into the scrotum. And that's happens in a fair amount of people, a fair amount of men, so it's not uncommon for people to have hernias repaired. But at any rate, the spermatic cords provide a passageway into the scrotum for these important structures. All right, so here it is. Here we have a lot of layers here. So we're looking from the outside. Here's the scrotum we've cut away. We'll see lots of layers. We're going to start looking at the gamete first, the the gonads first. And in the male, that would be the testis, and the testis is this egg-shaped thing here. There's one on either side. We've got this thing called the epididymis. Then we're going to have the ductus deferens running up here. Oops. We're going to have the ductus deferens running up. And you can see where it's going to curve back around. And then we've got the tympaniform plexus here, which is basically a venous plexus. We've got the testicular artery, which is the gonadal artery here. The inguinal canal is right here. And you can see how that might be a weak spot for things to pass through. All right, then we're going to have several layers that we'll talk about in a little bit. We've got several muscle associated with this area. We've got a layer of skeletal muscle called the cremaster muscle, and we also have the dartos muscle, which is smooth muscle, which we'll talk about a little bit later. And we'll talk about some of these structures because they're important in regulating how close the testes are to the body. So these things are going to change. The tone in these muscles will change when the temperature changes. So if somebody jumps into a cold pool, the testis will be pulled up close to the body. So if you jump into a cold swimming pool, then that's likely to happen. And then if you're in a very, very hot environment, well, then there'll be some relaxation and the testis will be allowed to be further away from the body. And the reason for that is simply so that body temperature, or I should say testicular temperature, is regulated so that it's going to be the right temperature for optimal sperm production. All right, so the scrotum is the outer covering, and the penis is the erectile organ. That's the thing that hangs down in the front. So we're going to have the end of the urethra running through the penis. It's going to be called the spongy urethra because it's going to run through a structure called the corpus spongiosum. And this is basically a bunch of blood vessels. And there are 3 erectile bodies in the penis. There's the corpus spongiosum, and we also have 2 corpora cavernosa. And we'll talk about these. All of these are basically just areas where we have some blood vessels that when they become dilated, then the organ gets engorged. Now, the scrotum is going to hold the testis, and there's one on either side, and there's actually a septum between them. And that is sort of a safeguard so that if one gets infected, the infection doesn't spread to the other one. All right, so if we look at it in sagittal section, here we have the sagittal section. Here we can see the structures. Here we can see the testis. Here we see the epididymis. Then we see the ductus deferens. We see it come up, loop over the bladder, back behind it. We're going to see, here's the ejaculatory duct here. Here are the seminal vesicles. Here is the prostatic urethra. That's the part of the urethra. Here's our urinary bladder. The prostatic urethra is going to be the part that passes through the prostate, which is this gland right here. And let's pause for a second and let's talk a little bit about the urethra, because the urethra is going to be a passageway not only for sperm, but also for urine. So this is going to serve 2 functions. The urethra is going to do double duty. It's going to play a part in the urinary system in the male, as well as the reproductive system. So we have to have a passageway from the bladder to the distal urethra. And of course, the urethra passes through the prostate here. So this would be the prostatic urethra. But then we're going to have the seminal vesicles, and we're going to have if this came all the way around, you could see where it basically dumped into here, and we're going to have the ejaculatory duct here, and then that's where the sperm are going to join the urethra. So the sperm come out here, and the urine comes here, and they will share this passageway. Now, interestingly, if someone is in the process of sexual arousal, then this part will be shut off so that basically the urethra will not allow for the passage of urine while that they're having intercourse. So here we have the bulbourethral glands. These are some accessory glands we'll talk about a little bit later. All of this structure here is called the corpus spongiosum. And we have 2 corpora cavernosa here. And there's one on either side. These are on the superior surface, and the inferior surface we're going to have the corpus spongiosum, through which the urethra passes, and that's why we call it the spongy urethra. All right, anything else on this slide? I think that's pretty good. Here's our pubic symphysis to orient you. All right, now let's look in the back end. Okay, so here we can see the rectum. This is our seminal gland, and here we're going to have our ejaculatory duct. So it does show it on the next slide. And you'll notice here the proximity of the rectum to the prostate. So when someone has a prostate exam, it's very easy just to feel it through the rectum. Another important thing is if somebody is going to undergo radiotherapy for prostate cancer, for example, or be treated for prostate cancer, well, they first have to do a biopsy to see if it's in there. So they'll come through the anus, and they'll come through the rectum, and then they'll take core samples through here, usually 12 to 18 core samples, and they'll just bore a little core right out of the prostate and check it out to see if there's cancerous cells there. In fact, a lot of guys tend to get prostate cancer later in life. Most prostate cancers are not that aggressive, and if they're not aggressive, then they usually leave them be, because the person will outlast the cancer, so to speak. So by the time the person, or I should say the cancer, would outlast the person, so that by the time the cancer would actually become malignant, the person's already dead. Now, there are other forms of prostate cancer that are very aggressive, and they have to be dealt with immediately, because if they're not, then the person will probably die. So the prostate is another finicky little gland, in that as men age, some of the tissues will grow out into the urethra, and it'll start to occlude the urethra, and this becomes problematic because then it makes it very difficult for men to void or urinate, and that can become problematic in later life as well. And sometimes a procedure has to be done where they'll take a scope and they'll take a little, sometimes it's done with laser. There are many ways to ablate it, but they'll go in there and ablate some of that tissue surrounding the urethra to free up the urethra a little bit. All right, so the scrotum is divided into two chambers called scrotal cavities, and these are separated by by a septum, and then you have this thickening of the scrotal surface called the raphe, and that is going to mark on the surface sort of where the separation is, where each of those two scrotal chambers is. And the reason for having two separate chambers is in case you should have an infection or something like that, you have kind of a backup. You got a sort of a fail-safe to keep the infection from spreading over to the other testis, and of course there are two testes in case something should happen to one. All right, so surrounding the inside of the scrotum we have a serous membrane, and this is called the tunica vaginalis, and this is because it evaginated with the scrotum when it was formed, and this obviously is going to have a very similar function to any of our serous membranes. It's going to reduce friction between the opposing surfaces. So we have the parietal layer, which is going to be flush with the scrotum, and the visceral layer that's going to be next to the testis. We have a layer of smooth muscle called the dartos muscle, and we have the cremaster muscle, which we already discussed, and this is going to be a skeletal muscle that's going to be deep to the dermis, and it's going to tense the scrotum and pull the testis closer to the body when it's cold, because obviously you want to regulate the temperature within the scrotum within an optimal window. All right, temperature regulation, as we know, normal sperm development is lower than typical body temperature, so about 2 degrees Fahrenheit lower than typical body temperature. And this is why there are these two sets of muscles that can cause they can either relax and move the testis further away from the body, or contract and pull the testis up closer to the body and keep the temperature a little bit warmer in there. Another reflex that people have is if there's danger. So another time, if someone's fighting or someone's doing something athletic that might, make the testis more vulnerable, also sometimes there'll be contraction of these muscles pulling the testis closer to the body. All right, then we have something called the tunica albuginea, and this is a little bit deep. This is deeper than the tunica vaginalis, and this is a layer of collagen connective tissue, so we got a lot of collagen fibers in this connective tissue, and this is going to be continuous with the fibers surrounding the epididymis, which is going to be the first organ that sperm move into once they're made in the testis. So they'll mate in the testis, and then they'll travel to the epididymis, which has a head and a body and a tail, and then that will lead into the ductus deferens. And so then we're going to have these fibers that are going to form partitions within the testis called septa. And these are going to converge near the entrance. And we'll see how that works. There's a whole area. There's a mediastinum of the testis and a rete testis. We'll see these a little bit later on as we move through the structures. So this is going to help bring blood vessels and lymphatic vessels to the testis and the efferent ductules. Okay, here we see a little bit of the layers. Here's our cremaster muscle here. They've pulled the penis up so that you can see underneath. Here is that scrotal septum. Here would be the midline raphe. Here's the scrotal skin. Then you have the dartos muscle. Then you have the scrotal fascia, cremaster muscle. And on this side, you can see a little bit of this would be the tunica albuginea here. But the tunica vaginalis is not really shown here, which is our serous membrane. All right, let's see. All right, so if you look in the testis, that's where we're going to start first because this is where the gametes are formed. We're going to have these lobules, and these lobules have these really tightly coiled what we call seminiferous tubules, and these are going to produce the sperm. And each one of these is about 80 centimeters long, and if you were to string out all the seminiferous tubules, you'd get about a half mile of seminiferous tubules per testis. So here they are. Here are our lobules here, and you can see the seminiferous tubules that are all coiled up inside. And then we're going to have basically these, the rete testis here, which is going to be located within the mediastinum of the testis. And then we're going to have these efferent ductules. So basically all the lobules are going to empty out right here, and here's our rete testis. And then we're going to have our efferent ductules, which as the name implies, efferent, it's going to be leading away from the testis. Those will lead into the epididymis, which also has a bunch of tightly coiled tubes in it. And here we have our head of the epididymis, and then we have the body of the epididymis and the tail of the epididymis, which leads into the vas deferens. So some people will ask, well, is it ductus deferens, vas deferens? What do you want us to call it? Well, vas deferens is it made, right? So as long as you get one of the anatomically correct names, I'm not going to be really worried about it. All right, so this obviously is going to travel in the spermatic cord along with blood vessels and things. All right, so let's see. Here's a different view of it. Here we're looking sort of in coronal section here. Here you can see each of the lobules. Here we can see all the layers very nicely. We can see the septa that separate the lobules. We can see the mediastinum of the testis here, this part right here. We can see the rete testis here, and we have our efferent ductules leading out of the rete testis into the head of the epididymis. We can see the tunica albuginea here, which is basically going to infold to form these septa between the lobules. Then we have the tunica vaginalis. You can see the visceral layer here, and here you have the fluid space between, that very thin fluid, just like we have in other serous membranes. Not much of a space, really, just fluid-filled, very thin cavity there. And then you have the parietal layer of the tunica vaginalis right here. Then we have our cremaster muscle. And then from there, we're going to see our superficial scrotal fascia, and then the dartos muscle, and the skin. So that's pretty much a layered tour through the testis. And here you can see the seminiferous tubules. They will actually enter into the rete testes via this thing, which is called a straight tubule. So this part here is the rete testes, which resides within the mediastinum of the testis. So this network here is the rete testes, and that's going to be receiving sperm from these straight tubules from the actual lobules, so from the seminiferous tubules in each lobule. From there, it's going to travel through an efferent ductule right here into the epididymis. All right, so what are we going to find in the seminiferous tubules? Well, we're going to find sperm at various stages of development. And so we're going to have spermatogonia and spermatocytes. And spermatocytes will undergo a very special type of division called meiosis. And this is a reductive division. So basically, we want to take our genetic material and half it. Whenever we make a gamete, obviously, if we have 46 chromosomes, 23 came from the mother, 23 came from the father. So when we produce these gametes, we're going to half the amount of DNA that's in them, so that we're basically creating 23, or gametes with 23 chromosomes. And so meiosis is this kind of reductive division where we have cells divide, but they're only going to have half the DNA of the individual, of the organism that made them. So then we're going to have spermatids, which are kind of immature spermatozoa. And we're going to have these very large nurse cells. They're also called Sertoli cells. And these are going to be sort of overseeing the development of the spermatozoa through their development. All right, so we're going to see some major functions in these nurse cells. First of all, obviously gametes look really, really weird to the rest of the body. They only have 23 chromosomes, right? And they look kind of foreign. If there were going to be some, let's say, a resident macrophage or a T cell going through there, they'd be like, wow, this looks weird, I better attack it. So we have the blood testis barrier, and that's going to keep things out. That's going to keep the developing sperm separate from the rest of the body. We're also going to support mitosis and meiosis. Our spermatogonia are going to undergo mitosis because these are the germ cells to make the germ cells. So I should say the stem cells. So basically, they are going to be the ones that are going to divide, and they're going to be the creator of the germ cells. So basically, they're going to have 46 chromosomes, but then they're going to produce cells that will then divide and have 23 chromosomes each. And it's the division from that point on that we start what we call meiotic division. We're also going to secrete a hormone called inhibin, and as the name implies, that's going to inhibit some of the hormones that will be controlling the actions of the testes. Remember our pituitary, hypothalamic pituitary axis. That's going to be highly influential in both male and female. We're going to see a lot of hormones produced in the hypothalamus, we've already looked at them before, and the pituitary that are going to affect the reproductive function. So follicle stimulating hormone is a very important hormone that we talked about earlier, and it's going to promote the development of sperm. So follicle stimulating hormone will promote spermatic development. And we'll see later in the female, it's also going to promote the development of the oocyte. So FSH is basically a hormone that promotes gamete production. Then we're going to see gonadotropin releasing hormone, and that's going to obviously affect FSH and LH. And LH, we'll see in the male, is going to cause production of testosterone. In females, it has a slightly different function. We'll look at that later on. And we also have something called androgen binding protein, and that's going to concentrate the testosterones in this area, and that's going to promote spermatic development as well. All right, so let's see here. Here's our blood testis barrier. Here is a nurse cell, and here's the nucleus of the nurse cell. And then we've got, if we're looking from the inside, here's our connective tissue here. And then we have got our, what we call the basal compartment. And the basal compartment is going to have, this is going to be on the inside of the blood testis barrier. I should say, it's going to be on the part that's not really protected from, so it's going to be really maintaining the blood testis barrier. But the spermatogonia are here. And these are the ones that are going to undergo mitotic division. And then anything that undergoes meiotic division is going to be within the blood testis barrier and shielded from the rest of the body, so that, the body really doesn't see it. Because as we say, these are very odd looking cells to the rest of the body, and they would be very easily under attack very quickly if cells of the immune system saw them. All right, so we've got these spermatogonia here, and the spermatogonia are going to be making primary spermatocytes, and then primary spermatocytes will develop into secondary spermatocytes, which will then begin to develop into spermatids. And spermatids are incomplete, and as they become sperms, they will start they'll undergo a process called spermiogenesis, and that's when they'll start to develop their tails and all of that. Now, this whole process from bottom to top is called spermatogenesis. Spermiogenesis is the last part of that process when they mature from the spermatids into the spermatozoa. And spermiation is another process when they actually release from the wall and now they're free in the lumen. And we'll go through those processes in a little bit more detail in a bit. All right, now let's go back to the testis for a second. Let's look at some more structural details. We've got the connective tissue capsule that's going to surround the tubules. We've got areolar tissue that's filling the spaces between the tubules. And we have got basically these blood vessels. But we also have interstitial cells. These are also called the interstitial cells of Leydig. And these are going to be producing our androgens, testosterone being one of the more dominant or the most important of these sex hormones. All right, so let's have a look. Here is our histological thing. Here we see some maturing spermatozoa here. This is the lumen. This is where they're going to get released into. We've got spermatogonia here. Here's a nurse cell or Sertoli cell. Sometimes it's called that as well. These interstitial cells, also known as the interstitial cells of Leydig. Then we've got some dividing spermatocytes. We'll go more into the details of meiosis a little bit later on. I'm not going to make that a prime focus, really. We'll just get into the basics of it. But for now, just suffice it to say that the meiosis is going to be a reductive division that's going to convert cells that originally had 46 chromosomes, the spermatogonia, and from there we're going to divide, and we're going to have two cells that will have, well, from this phase forward, we're going to have 23 chromosomes. So this is the important thing, is we're reducing the number of chromosomes to half, and that's what meiosis is all about, and it is for the production of these germ cells. All right, so spermatogenesis, that is the whole entire process from beginning to end of sperm production. And so that's going to begin at that outermost cell layer in the seminiferous tubules and proceed toward the lumen. So from the spermatogonia to the lumen, that's the process. So we're going to start from the outside and work our way in. All right, so our spermatogonia, these are our stem cells, and they're going to divide by mitosis. They're going to produce two daughter cells. One is going to stay a spermatogonium because we have to have some way to keep producing sperm, right? The second one is going to differentiate and become a primary spermatocyte. It is the primary spermatocyte that's going to begin meiosis, and that's this reductive division to 23 chromosomes. And this is going to form a secondary spermatocyte. From there, we're going to have the secondary spermatocytes are going to differentiate into these spermatids, which are immature gametes. They don't yet have the tail and everything. They're just getting ready to become sperm, and they will differentiate in spermatozoa. And the spermatozoa, once they have all of their parts, they've got their head, they've got their middle piece, they've got their tail, then they will actually break away from the wall of the seminiferous tubule and enter the fluid in the lumen. Now, they will not be motile. Their little tails will not be swimming. And the reason for this is they've got to wait till delivery, because that's when they're going to need to use the tail, and so they're not really going to become motile until much later. And that will actually be part of a process we call capacitation, which we'll talk about a little bit later. All right, so spermatogenesis has three integrated processes. Mitosis, which is that first division of the spermatogonia, one that will stay a spermatogonia, one that will become a primary spermatocyte. Then meiosis, which is begun by the primary spermatocyte, and that will end in spermiogenesis. And spermiogenesis is really that final maturation step. And then we'll have spermiation, boom, when they release the wall and they're in the lumen. All right, so during mitosis, we're going to have this somatic cell, that's a cell of the body that has 46 chromosomes, is going to produce two identical, what we call diploid daughter cells. What does diploid mean? We have 46 chromosomes, so they both will have 46 chromosomes. When we get to meiosis, then when we have one set, we'll have 23 chromosomes. We call these haploid cells. All right, these are going to be used for cellular regeneration, very important here, because we need to be able to make more of them down the road. All right, meiosis. That is our special form of cell division, and it's only in the production of gametes, so spermatozoa in males. So remember, each gamete has only 23 chromosomes, because eventually, if we have conception, they're going to fuse together. The gametes will fuse, each has 23, and they'll produce a new offspring with 46 chromosomes, 23 from the mother, 23 from the father. All right. So in the seminiferous tubules, we're going to have our primary spermatocytes, and these will eventually produce our spermatids. All right, so spermiogenesis is the last step in spermatogenesis, and that's when we have our spermatids. These are relatively unspecialized yet. They don't have their tails, but they have already undergone meiosis. And then we're going to have these spermatids differentiate into sperm. So these are our highly specialized cells. We will see that the only structures that they carry, they've got a head that carries the DNA, and then they have all the machinery to make the tail go. And that's basically it, because these are just little delivery vehicles for DNA. And so we don't have any other really cellular organs except those that are like mitochondria that are important for getting this thing to swim and take the DNA where it's supposed to go. So it doesn't have the cellular structures that we'd see in most other cells. These are highly specialized delivery vehicles. All right, so here is our spermatid, and watch what happens. We've got our Golgi apparatus here. This is a relatively unspecialized cell yet. The Golgi apparatus will start to form an acrosomal vessel. And this is going to be important because it's going to be full of enzymes. And this is going to sit at the head of the sperm, so that when it meets the egg, it's going to be able to break through that outer covering and get in to deliver the DNA. So that's the whole point of the acrosome. So we have this acrosome that's sitting on the head, and that's kind of like the way to get in. It's kind of like the explosive, it's the enzymes that are going to allow the sperm access to the egg. So that's kind of like the explosive head on a bomb, if you will, or a missile. So the acrosome then is going to be forming from the Golgi apparatus in the spermatid. And the acrosomal vessel will start to form from the Golgi apparatus until it becomes the acrosome. And then we will have, basically, the DNA condensed down into this head, this nucleus, which is going to have the DNA in it. And then we're going to have this sort of middle piece here that's going to have a lot of mitochondria in it. And then we're going to have the tail. And we're going to be losing cytoplasm all along because we're not going to need it. We're just going to get this thing down to its most functional thing, this swimming thing. Spermiation is when the spermatozoa loses attachment to the nurse cell. Now remember, the nurse cell, also known as the Sertoli cell, is going to be overseeing the process of sperm development all the way through from basically the beginning, the primary spermatocyte, up to spermiation. And spermiation is simply when the sperm loses its attachment, and now it's in the lumen of the seminiferous tubule. So this whole process, from the first division of the spermatogonia that's going to lead to this gamete, takes about 9 weeks. So this is constantly ongoing. All right, so when we look at the mature spermatozoon, as we said, it doesn't have a lot of the cellular structures that we would see in other cells. So, for example, we don't have an endoplasmic reticulum. We don't have our Golgi apparatus. That basically formed our acrosome. We don't have lysosomes or peroxisomes or other cellular structures. And so this really makes it, as I say, a delivery vessel. It doesn't even contain its own energy stores. What it's going to do is it's going to absorb fructose. And we'll see that fructose will be produced, especially later on in this process, fructose will be produced by some of these accessory glands that will be secreting things into the fluid all along. So we're going to have all along this journey, we're going to have these accessory glands that will be secreting fluids into the lumen and supporting the sperm. All right, so if we look at a spermatozoon, we got a head, a neck, a middle piece, and a tail. So let's look at those things. The head has the nucleus, which has the chromosomes, and this acrosome, which is a cat-like compartment. And basically, this is enzymes that are going to help the sperm get through, and it was derived from the Golgi apparatus. Then we got our middle piece. This is going to attach basically the head to the tail via a neck. That's going to have our mitochondria in it, which is going to power the thing, and we're going to see this sort of spiraling around in these spiral around microtubules that will be the base of the tail. So that's going to provide the energy to move the tail, and the tail will whip around in this corkscrew-like pattern. And the tail is a flagellum, and it's the only flagellum that we'll ever see in the human body, and basically it is for motility. All right, so here it is. Here is our nucleus. The acrosome surrounds it. All of this is the head. Then here we have our centrioles. This is our neck. This is our middle piece full of mitochondria. And then we have the tail. And this is about what they look like here. So you can see the head through the neck and the middle piece and the tail. All right. So once the spermatozoa is getting fairly close to being ready to be released, so spermiation, this is when it's ready to be released and it detaches from the nurse cell. Now it's floating freely in the seminiferous tubules, and it will not start to swim yet. It's incapable of locomotion or fertilization. This is a step we call capacitation, and it's basically a two-step process, and we'll study that a little bit later. But basically, it will be moved along by cilia. Remember when we talked about cilia being cells, or cells that have cilia are specialized for moving fluids across surfaces or in the lumen of some of these tubular organs. So the epididymis is lined with cilia, and so we have the seminiferous tubules also are lined with cilia. So we're going to see cilia all through the reproductive tract. So the seminiferous tubules are where these things are being produced, and then we're going to have the cilia lining the ductules are going to move the sperm into the epididymis. And the epididymis, as we saw, has the head sitting on top of the testis, and the body and then the tail at the bottom, which curves back up to run into the ductus deferens. And this is the start of the actual tract. This is the tubular part. And so we have coiled tubes here that are about 20 feet long, 23 feet long. And let's see if we got a good picture of it. This is mostly, here it is. Here is our head here. Here's our body, and here's our tail. And here it runs in the ductus deferens. So once again, here we have our testis. We have our tunica albuginea, which makes these little septa that separate the lobules, and the seminiferous tubules are in the lobules. We have the straight tubules leading into the rete testis, which then we have our efferent ductules leaving the rete testis, going into the epididymis. Here is the head of the epididymis, the body of the epididymis, and the tail of the epididymis. And then the next stop along the way, actually they don't really, well, the next part of our journey will be up the ductus deferens. All right, so here are cilia. Here you can see them. So here you can see some spermatozoa up here in the lumen of the epididymis. And here we have pseudostratified columnar epithelium of the epididymis, and you can see the cilia. All right, so what does the epididymis do? Well, one of the things is going to adjust the fluid produced by the seminiferous tubules. It will also recycle damaged spermatozoa. So some of them don't quite make it out of this process. And so it's going to recycle the damaged one, and it's going to store the others, the ones that are going to move on to whatever, greater things. So this is going to facilitate functional maturation. Right now, they're physically ready, but they're not mature. They're not swimming yet. All right, so basically they're mature, but they're immobile. And in order to become motile and swimming, they're going to have to undergo capacitation, which we talked about earlier. And part of capacitation is for them to become motile. And as we said before, it's 2 steps. The first step is when they become motile. This is when they will be mixed with secretions of the seminal glands, and these will be fructose-rich secretions. This will provide them the energy that they need. And then they become capable of fertilization only when exposed to the female reproductive tract, and that's step 2 in capacitation. All right, so let's look at the vas deferens, or the ductus deferens, and it's about 16 to 18 inches long. As we said, it begins at the tail of the epididymis and basically ascends through the inguinal canal, curves along the urinary bladder toward the prostate glands and the seminal glands. There's only one prostate gland. It's right in the middle. It's right under the bladder. And then the lumen's going to enter into this large ampulla. It's going to have a thick layer of smooth muscle there. All right, so let's see it. Here we have our ductus deferens coming in. Here's our ampulla of the ductus deferens. Here are our seminal glands here. And then we're going to have the duct of the seminal gland that's going to empty into the ejaculatory duct, which is where we're going to get the sperm coming in. And then we're going to have the prostatic urethra here. And then we have our membranous urethra. This is where we have our urogenital diaphragm. I'll point out our bulbourethral glands again. And then we're going to have the spongy urethra here. And this is the part that's actually in the penis. All right, so this is what it looks like. This is the duct of the ductus deferens. So this is the lumen. We've got a bunch of smooth muscle around that. All right. So the ductus deferens also has a ciliated epithelium. And we also have some peristalsis that's going to propel the spermatozoa in their fluid towards the ejaculatory duct. Now, the sperm can stay in here for several months, and they're very low metabolic rates. You can sort of think of them in suspended animation, waiting for their day. All right, and the ejaculatory duct is just simply a short passageway at the junction of the ampulla with the seminal duct. And this is going to penetrate the wall of the prostate and empty into the urethra. All right, so we got a bunch of accessory glands as well, and these are producing semen. Semen is obviously going to be the mixture of the secretion from all these different glands, and they're going to have different characteristics. So we're going to have seminal glands, prostate glands, bulbourethral glands, and they'll each contribute to the composition of semen, and semen obviously is the vehicle by which the sperm get into the female reproductive tract, and it also contains things that are going to be essential to the survival of the sperm and also facilitate them being able to do their job. All right, so typically if there is an ejaculation, it's about 2 to 5 milliliters of sperm, and there could be any number of things along the way that might cause problems here. For example, if your testes are too close to your body, you might have low sperm count. You could have prostate glands or seminal glands not doing their job and producing the fluid. So typically when someone has a sperm count done, they're going to wait 36 hours after sex, obviously, because you don't want to deplete your stores. You want to see how many you're making. And typically 20 to 100 million sperm are what you would find in 1 milliliter of this fluid. So when people have sperm counts done to see what their fertility is, then they'll basically do this test. They'll wait 36 hours, and they'll go and collect the semen and see how much spermatozoa is in it. All right, so the ejaculate is obviously this fluid produced by an ejaculation. So it's got seminal fluid, spermatozoa, and enzymes. We got lots of enzymes in here. We got protease, which is a proteolytic enzyme. We've got seminal plasmin, prostatic enzyme, and fibrinolysis. We'll see why that's important, because to travel, a lot of times these things will travel in a clot. So sperm will clot together, be shot through like a cannonball, and then they'll break out into their individual sperm as they start to swim toward their target. All right, so obviously we've got some major functions of the male accessory glands, one of which is we've got to activate the spermatozoa. We also have to provide the nutrients for the spermatozoa. So we're going to be looking at fructose here. We're going to be propelling spermatozoa and fluids along the reproductive tract So we're going to be having peristaltic contractions all through here, and we're going to produce buffers, because remember, when they go into the female reproductive system, there's going to be, and in their own reproductive system, there's going to be acidity. Think about the urethra and what passes through that. So the male urethra can be acidic, and then it's going to pass into the vagina, which will have its own environment, and obviously these sperm have to be buffered against anything that would be harsh. So basically it sort of makes buffers so that the sperm can more easily survive. All right, so seminal glands. These are the ones that the two glands that are nestled inferior right behind the bladder there. And so it's about 6 inches long, and it's tubular gland, and basically it's going to produce about 60% of the seminal volume. So if we look at it, here it is right here, seminal gland, and it's going to be dumping its secretions into the ejaculatory duct along with sperm, which are coming from the ampulla of the ductus deferens here. All right, so seminal fluid has about the same osmotic concentration of blood plasma, but a completely different concentration. As we know, we've already talked about the fact that fructose is needed by the spermatozoa for energy. So we also have prostaglandins, and prostaglandins are going to stimulate smooth muscle contraction, both in male and female reproductive tracts. Fibrogen is going to form a temporary clot. As I said, these things are going to travel together, kind of in a cannonball kind of thing, and then it's going to be basically so that they can go in one package into the female reproductive tract. And we're going to have some alkalinity to this fluid, and that's going to help neutralize the acids in the prostate gland, vagina, and so forth. This is also going to begin our first step in capacitation. As we said, capacitation is 2 steps. The first step is when they become motile. So this is when their flagella will start beating, and they're going to start swimming. All right, then they're discharged into the ejaculatory duct at what we call emission. And emission is basically when they're in the ejaculatory duct, when they get put in there, and we have some peristaltic contractions pushing them there. And these contractions will be controlled by the sympathetic nervous system. So interestingly, when there is coitus, there are going to be both sympathetic and parasympathetic involvement here. So unlike maybe some other organs where we see a dual innervation, and we see other organ systems, we'll see kind of a antagonistic interplay. Here we have an interplay that's more one than the other, like they're handing it off. So basically the sympathetic stimulation, the sympathetic nervous system, is when the person's getting stimulated and aroused and all this is about to happen. And then later we'll see when the parasympathetic nervous system kicks in. All right, here's our lumen of our seminal gland. You can see all of these basically glands, these cells, these ducts that are going to be making up this gland. And we're going to have some smooth muscle, of course. All right, then we're going to have our prostate gland. This is the organ that sits right under the bladder, and it's going to encircle the proximal portion of the urethra, and hence we call it the prostatic urethra, the part of the urethra that passes through it. And it has a bunch of tubuloalveolar glands and surrounded by smooth muscle, as we saw in the previous slide. Here it is. Let's see if I can get my cursor on it. Here it is right here. This is the one that sometimes starts proliferating later in life, and you get prostatic tissue into the urethra, and it blocks the urethra and can cause problems. So a lot of men will eventually have a little catheter put up here, or I should say they get a well, they'll come out here and remount some of this tissue. Most men don't need a complete prostatectomy, which would be taking the thing out. They do that in certain cancers, but they do. A lot of men, as they reach 60s and 70s, will have some of this prostatic tissue in here removed, because it does tend to grow into the urethra. All right, so here is some of the prostate gland from a histological view. Here we have our lumen inside of here, and you can see these glands. This is actually the lumen of the glands themselves, and obviously some connective tissue and smooth muscle in here. All right, so here we have a slightly acidic fluid. This is going to be about 20 to 30% of seminal volume, and this is going to contain an antibiotic called seminal plasmin. And this is going to be ejected into the prostatic urethra by peristalsis of the prostate wall. So the prostate wall, obviously, is what's lining, or I should say forms the wall of the urethra which runs through it. So, all right. Now let's look at the bulbourethral glands. These are going to be right there at the membrane, at the urogenital membrane there. And so these are going to secrete an alkaline mucus, and this is going to help neutralize acids in the urethra. It's also going to lubricate the glans penis, which is the very tip of the penis. It's the thing that kind of looks like a mushroom in the front. All right, so each of these glands is going to be alongside the penile urethra, and that's the part that goes through the penis, and that's the part that travels through what we call the corpus spongiosum. All right, we'll talk about that when we get to the penis in just a little bit. Here are our bulbourethral glands, and here's our urogenital diaphragm. All right, so here's our bulbourethral glands. Here you can see smooth muscle again. You can see the glands. You can see the lumen inside. All right, now the urethra. As we've said before, it's used by both the urinary and the reproductive system. So when reproductive function starts, basically the urethra is shut down to urine. So basically shuts that pathway down. And basically it's about 7 to 8 inches, depends, from the bladder to the tip of the penis. And as we said, it's divided into 3 regions. Our prostatic, that's the part that passes through the prostate. Our membranous, which is the part that passes through that urogenital diaphragm or membrane there, a lot of muscle there. And then our spongy urethra, which is going to pass through a structure called the corpus spongiosum within the penis. And it's one of 3 chambers that are erectile chambers. They're filled with blood vessels that will dilate and become engorged. All right. So the male external genitalia is the penis. That's that tubular organ. And that's the one that the distal part of the urethra passes through. And not only does it allow for voiding and conducting urine to the exterior, but it's also going to be the part that, if there is coitus, is going to be the part that goes into the female reproductive system. All right, it has a root at the bottom. That's going to attach the penis to the body wall. And it's going to occur with an area called the urogenital triangle. We'll see that. That's right below the pubic symphysis. It has the body, which is the shaft. That's the tubular part. That's the part that can move. And the glands, which is that distal tip, that thing that looks kind of like a mushroom. All right, so if we look at it here we have our urogenital diaphragm. So our membranous urethra is right here. And remember, all this is prostatic urethra. This is our bladder. So we got our bladder and our prostatic urethra, membranous urethra, and then we have the spongy urethra moving through here. Here is our glands. There's a covering called the prepuce. This is often what is removed when at birth. A lot of times people remove this at birth. All right, so if we do a cross section through it, that would be this little thing right here. If we do a cross section through the penis, we can see our erectile tissues here. Here is our corpus spongiosum with the urethra in it. These are the 2 corpora cavernosi, and these are also basically blood vessels that will become engorged. This is on the dorsal surface, or that surface on the top. And then we're going to have some arteries for the penis here. We've got arteries in the corpus spongiosum here. We've got, corpora cavernosa. We've got 2 corpora cavernosa. Each one would be a corpus cavernosum. And we got a lot of blood vessels. Here you can see, here's the sheath of the penis. It's a collagenous sheath. And then we're also going to have a part of the dartos muscle that's continuous from the scrotum to the penis. This gives it that crinkly appearance. This is why the skin covering these areas looks somewhat crinkly, because of the tension that's going to be in the dartos muscle. And then here we have the skin.